Hello class, this is chapter 5 part 2. We're going to be talking about the three types of passive transport across membranes. So we need to talk about solutions. A solution is it's a mixture usually of a solid and a liquid. And my favorite example of solutions are either something like Kool-Aid or Crystal Light. So you're trying to make a drink, either the Kool-Aid or the Crystal Light, and that is your solution, the final product. And it consists of a liquid, which is water, and a solute, which is the powder in your packet. So you open up your packet, you put that powder into some sort of container, then you add the right amount of liquid and mix it, and you get a solution. So the liquid part, the water, is called the solvent. It is the thing that dissolves the other thing. So it's the substance that does the dissolving. The act of dissolving is that powder disappearing into the liquid. It is mixing with the liquid to a point where you can't tell they're two separate things. You know, at the end of making your Kool-Aid or whatever, there is typically a couple particles down there. Some of that is usually part of the packaging, actually, but some of it is some larger bits of sugar and flavoring. But basically, for most of that powder, you can't, see it within the liquid. It's just changed the water a different color. And that's sort of evidence that they've truly mixed and become one solution. So the solvent, the thing that does the dissolving, is usually the liquid, and if it's in biology, it's usually water. The solute is the Kool-Aid or Crystal Light packet. So it's not the packet itself, but the powder in that packet. That's the solute. And it is the thing that is dissolved. So the solute is dissolved, the solvent does the dissolving, and then once the dissolving is done, you have the solution. So that's the first part we need to know. We also need to know diffusion. And diffusion is the movement of something down a concentration gradient. A gradient just means a difference. And typically, we're comparing the inside and the outside of a cell. Hopefully I spelled that right. I'm not sure I did. Anyway, so the gradient means there's a difference, and the thing that's different is the concentration. Concentration is a measurement of how much solid there is in a certain amount of liquid. And the concentration is predetermined for you in Kool-Aid, but you could, double the concentration by adding a second packet, right? Or you could have the concentration by using half of a packet. So when we're When we're making it more concentrated, we typically say we're concentrating it. When we're making it less concentrated, we're typically diluting it, right? So diffusion will happen from an area of high concentration where it is concentrated to an area of low concentration where it's more diluted. And that movement from high to low is just to the fact that molecules like to spread out and sort of be in balance and equilibrium. So think if I put like a whole classroom of students in one corner, y'all would be bumping into each other and not feel very comfortable, and you would want to spread out. You would want to use all the space of the room. And molecules do that too, basically for the same reason, because when they bump into each other, they go in opposite directions due to physics. And so they will naturally spread out due to those collisions. Some things that can affect diffusion is temperature. So when temperatures are higher, diffusion happens faster, it's more active. When temperatures are lower, it happens slower. When things are large, they diffuse slower than if something was smaller. So smaller molecules tend to diffuse a little bit faster than larger things. But the biggest determinant is the concentration difference. How big is that difference? How large is that gradient? That is typically the main driver of the rate of diffusion. And diffusion will happen until you hit equilibrium. Cells like to take advantage of diffusion, so they typically don't allow things to hit equilibrium. And there's different ways they can rig it. But let's think about putting a drop or a cube of dye into a liquid. When it first enters that liquid, it's at the highest concentration. That dye actually excludes water for a second, and then there will be some interactions. And water will go in, and the dye will go out. Each molecule type will diffuse on its own. So water will diffuse in a different direction than the dye. And that's because there's more water out here and there's more dye in here. And so they'll go where there's less of it. And so this picture just shows what I just said, which is that the water will move in to where the dye is, and the dye will move out to where the water is, until there's equilibrium and you have equal spacing of both water and the dye. And that's diffusion. Things will go from an area where they are concentrated towards an area where they are less concentrated. They will move down their concentration gradient. So over here the concentration is high. Over here it's low. We use that to our advantage with breathing. The air we breathe in has a high concentration of oxygen, and our blood has a low concentration of oxygen. So oxygen will move into our blood. Similarly with CO2, I can't really see that. There's a high concentration of CO2 in our blood, and there's a low concentration of CO2 in the air. So CO2 will move into the air, and then we can breathe it out, both by diffusion. The reason they can diffuse across membranes, because if you look closely, there's a cell there and a cell here. So there's 2 layers of cells here. And those gases can cross those plasma membranes without any help. And that is because they're small and they are non-polar. An oxygen double bound to another oxygen. They pull on electrons the same because they are the same atom. So this bond is non-polar, and it's just 2 atoms, so it's small, so it can cross the membrane as much as it wants, typically dependent on its concentration. So remember how I said how the body will rig diffusion so that it's always going in one direction. So we'll keep breathing in more air, and that will replenish the oxygen in our lungs. And so this is always high. And then in our cells will use up the oxygen from our blood. So the oxygen concentration in our blood is always low. And that way there's always a concentration gradient where oxygen wants to move into our blood. Pretty cool. Okay, so that was simple diffusion. Very few things can do simple diffusion where they cross a membrane on their own without any help. Those things are typically small and nonpolar, and the best examples are oxygen and carbon dioxide gas. But another example would be like a small fatty acid. Or cholesterol. Cholesterol is another good example. Most things use passive transport called facilitated transport. And with facilitated transport, you need something to facilitate the crossing of the membrane. That thing is usually a protein. It still uses diffusion to drive the movement, the direction of movement. So they will move down the concentration gradient from high concentration to low concentration. Because they're going downhill, down the concentration gradient, no energy is required. And it typically requires a channel protein or a carrier protein. And that will allow some sort of cargo to move into the cell. It still follows its diffusion gradient, so notice there's a bunch of these on the outside and 0 on the inside. Because of that, they will move into the cell. But this protein allows the movement, so it is facilitated diffusion. This is not simple diffusion because it requires this protein. If this protein was not here, the movement would not happen because it cannot cross the membrane on its own. So when I was explaining carrier proteins, I said they were a little bit more interactive. So they will physically bind to their cargo and move it into the cell. And so you can see that happening here. But again, this direction of movement is driven by diffusion, so no ATP, no energy is required. So that was facilitated diffusion. It's a lot like simple diffusion, but with simple diffusion, these molecules can cross without a protein. And with facilitated diffusion, it requires a protein. So that's the difference there. But their direction of movement is always going to be from high to low concentrations. So it's driven by diffusion and no energy is required. Our last mode of passive transport is osmosis. And I want you to repeat after me, osmosis is not always good for our cells. It is the movement of water, specifically the diffusion of water across a semi-permeable membrane. So this is the definition of osmosis. And diffusion hints that water is going to be moving from a high to low water concentration. So water will move from where it's highly concentrated to where it's lowly concentrated. The water concentration is opposite of the solute concentration. So if there's lots of solute, there's very little water. And if there's lots of If there's lots of water, there's very little solute. And it's basically a space thing. If you're sitting on the couch, your dog cannot sit there. Your dog or your cat. So where there's a lot of solute, there's very little water. Where there's a lot of water, there's very little solute. The reason osmosis happens is this right here. So water can cross the membrane through aquaporin, but the solute cannot. So osmosis will happen when the solute cannot move. Water will move instead to try to balance out the concentration. And water will always move to where there's more solute. So our movement of water will be to where there's lots of solute. Water is going to try to dilute that solute. What else did I want to say? Oh yeah, so water moves freely through the membrane through facilitated diffusion. But it's osmosis when the direction of its movement is due to the fact that a solute cannot cross. So let's look at these thistle tubes. At the bottom of this tube is a membrane that will allow small things to pass like water, but not larger things like, well, when we usually do this, it's usually sugar water. And we typically use table sugar. And what kind of saccharide is table sugar? It is a disaccharide, which is somewhat large. It's larger than a monosaccharide. And so our sugar is typically colored, and it cannot cross our membrane. But the water can, because it's small and it can cross this membrane. So they have submerged this thistle tube with the membrane at the end into a more dilute sugar solution. So I want you to look at these concentrations. And tell me which is higher. The higher one is the 10% one, because it's bigger than 5. And that 10% is talking about the solute. And remember, water moves towards where there's more solute. So water will move from the beaker water, which is 5%, through that membrane into the thistle tube, which is 10%. As water moves into our thistle tube, this level of liquid will rise. And the rising of that level of liquid is the evidence of osmosis. So let's go over some terms. Our 10% solution is hypertonic. And when you think of a hyperactive kid, they are typically more active, at least more active than what you want. They're a little manic. So hypertonic is more concentrated. That tonicity, it doesn't literally mean this, but I think of it as strength or concentration. So hypertonic solution is more concentrated. And hyper does literally mean more or higher. Our 5% solution is hypotonic. And if you think hypotonic, if you think of a hypodermic needle, those go under your skin. And so hypotonic is under or less or lower than the hypertonic side. And so if one side's hypertonic, the other side is hypotonic. If one side's hypotonic, the other side is hypertonic. And so if we look at the beginning of our experiment, the 5% was hypo. The 10% was hyper, and you can see that by the number of green spheres. Water during osmosis will always go towards the hypertonic compartment. And that's because where there's more solute, there is less water. So water will diffuse down its gradient to the hypertonic compartment and dilute out that sugar. Depending on your difference, you may never hit equilibrium. But ideally, water would continue to move into the thistle tube until these percentages equaled out. When things are equal, they are isotonic. And so iso means the same. So an isolateral triangle has the same length of sides and the same angles. And with osmosis, they're isotonic, so they would have equal concentrations. If one side is isotonic, the other side is isotonic. And water will move, but it'll move in both directions, so there'll be no change. So let's talk about cells in these different environments, and I recommend drawing a picture. You can do a circle for animal cell, a rectangle for a plant cell, and then usually we'll tell you what the solution is. So we'll say we put an animal cell in a hypotonic solution. That means since the outside's hypotonic, the inside is hyper. If you wanted to shorten this, you just put ER and O. Water always goes towards the hypertonic compartment, so water will enter the cell. That will make it swell up, and if the difference is big enough, it could burst. And another term for burst is lysis. Lys means to break or to burst. Some examples of hypotonic solutions is like distilled water. That is hypotonic. So if you put distilled water into an IV, you would definitely make the blood cells, the red blood cells in the blood, swell up, and they could possibly burst. So this is why we care, because you could seriously injure someone, or worse. If you put a cell in a hypertonic environment, that means the cell is hypotonic. Water will move towards the hypertonic side. So water would move out of our cell, and that will cause the cell to shrivel. If you put an animal cell in an isotonic environment, that means the cell is isotonic, and water will move in and out, but there will be no change to the cell. So I told you examples of a hypotonic solution that's like distilled water. Hypertonic would be like soda or seawater, and that's why we can't drink seawater. Seawater is worse than soda, obviously, because if you drink seawater, that's extremely hypertonic to your cells. Your cells would shrivel, and that makes them just as equally unhappy as bursting or lysing. An example of an isotonic solution is like contact solution or saline. And so if you had to give someone fluids in an IV, you would want to give them saline or something similar. That is isotonic. Plants are a little different, and it's mostly due to two structures, the central vacuole and the cell wall. Because of this, they can handle a hypotonic solution. So remember, in the animal cell, in a hypotonic solution, water entered our cell, the cell swelled up. And if it's bad enough, if it's hypotonic enough, the cell can burst. Well, a plant cell has a cell wall that can prevent the cell from bursting, and then the central vacuole can absorb excess water. And plant cells are actually happiest with hypotonic solutions. That's why we tend to feed them water. This water that enters the cell puts pressure on the cell wall. And water will still enter our cell. So if you put a plant cell, again, let's represent it by a rectangle, in a hypotonic environment, the cell is hyper, water will still rush into the cell. So the direction is the same. But with animal cells, they could swell and burst. With plant cells, they don't burst. Instead, they become turgid, or they have turgor pressure. And plant cells like being turgid. It's how plants without bark stand up. The reason plants without bark can stand up and be upright is due to this turgor pressure. So that's why we typically feed plants tap water or some sort of water solution. If you put a plant cell in a hypertonic solution, something very similar happens. So we'll draw a new plant cell. We'll put it in a hypertonic environment. This would be like salt water or Gatorade or soda. The plant cell would be in the hypotonic side. Because the outside is hyper, it's higher. That means the cell would be lower. Water will move towards the hypertonic side, so out of the cell. And that will cause our cell to shrivel. When an animal cell shrivels, we call it crenation. When a plant cell shrivels, we call it plasmolysis. So know the difference between those terms. Crenation goes with animal cells, plasmolysis goes with plant cells. And the reason it's a different term is because of the cell wall again. You can see the plasma membrane pulling away from the cell wall. Also in a microscope, you can often see the central vacuole shrinking. So with animal and plant cells in a hypertonic solution, the movement's the same, the shriveling is the same. We just call it slightly different things due to the fact that there's a cell wall in plants and not in animals. Alright, the last one is isotonic. The main thing with isotonic solution, plant cells are physically okay, but they lose the turgid pressure, so they're known as flaccid. And the plant may not be completely upright because it's losing that turgid pressure. The cell is still technically okay, even in an isotonic solution like saline. But, you know, without that plant being upright and erect, it's harder for it to collect as much sun as it can. And so it'll do less photosynthesis, and then the plant will suffer. So when you're comparing animal to plant cells in osmosis, the direction is the same. So isotonic, the water will move in and out, but there'll be no change. Hypotonic, water will move into the cell. And a hypertonic solution, water will leave the cell. So that direction is the same. But due to the cell wall, there are some differences. An animal cell in a hypotonic solution could burst. A plant cell will not burst. In fact, it is turgid, and that turgid pressure makes a plant pretty happy. In a hypertonic solution, both cells will shrivel. In animal cells, we call that crenation. In plant cells, we call that plasmolysis. That special term, plasmolysis, is referring to the plasma membrane pulling away from the cell wall. In an isotonic solution, there'll be no movement. Animal cells are happy. Plant cells are a little sad because they have lost that turgidity, that turgid pressure, and so they are flaccid. And plants without bark may not be upright, they may be slightly wilted. So know what happens to animal and plant cells in each type of solution. Be able to predict the movement of water. And remember, water will always go towards the hypertonic compartment. So you always want to label your compartments. So always label the environment and always label the cell. And then you can predict movement. So that's it for passive transport. I know that lecture was a little long. If you need to watch it again, make sure you take breaks between the facilitated transport and the osmosis.